Hi guys, today we will see acid-fast staining method for identification of bacteria. I have already posted videos on simple staining method and gram staining. You can get the link in the description box. So, acid-fast stain. Starting with the principle. A few species of bacteria in the genera Mycobacterium and Nocardia, and the parasite Cryptosporidium do not readily stain with simple stains. However, these microorganisms can be stained by heating them with carbol fusion. The heat drives the stain into the cells. Once the microorganisms have taken up the carbol fusion, they are not easily decolorized by acid alcohol, and hence, are termed acid-fast. This acid-fastness is due to the high lipid content, that is, mycolic acid, in the cell wall of these microorganisms. The Ziehl-Neelsen acid-fast staining procedure developed by Franz Ziehl, a German bacteriologist, and Friedrich Nielsen, a German pathologist. It is a very useful differential staining technique that makes use of this difference in retention of carbol fusion. Acid-fast microorganisms will retain this dye and appear red. Microorganisms that are not acid-fast will appear blue or brown due to the counterstaining with methylene blue, even after they have been decolorized by the acid alcohol. A modification of this procedure that employs a wetting agent, such as turgidal number 7, rather than heat to ensure stain penetration is known as the Keogh staining procedure. This procedure is developed by Joseph Keogh, German bacteriologist. Now, let's see the procedure step by step. Ziehl-Neelsen or hot stain procedure. Prepare a smear. Allow the smear to air dry and then heat fix. Place the slide on a hot plate that is within a chemical hood, and cover the smear with a piece of paper toweling that has been cut to the same size as the microscope slide. And saturate the paper with Ziehl's carbol fusion. Heat for 3 to 5 minutes. Remove the slide, let it cool, and rinse with water for 30 seconds. Decolorize by adding acid alcohol, drop by drop, until the slide remains only slightly pink. This requires 10 to 30 seconds and must be done carefully. Rinse with water for 5 seconds. Counterstain with alkaline methylene blue for about 2 minutes. Rinse with water for 30 seconds. Blot dry with bibulous paper. Acid-fast organisms stain red, the background, and other organisms stain blue or brown. See this figure for an example of the Ziehl-Neelsen stain. Keogh or cold stain procedure. This method may be used instead of, or, in addition to the Ziehl-Neelsen procedure. Heat fix the slide as previously directed. Flood the slide for 5 minutes with carbol fusion prepared with turgidal number 7. Decolorize with acid alcohol and wash with tap water. Repeat this step until no more color runs off the slide. Counterstain with alkaline methylene blue for 2 minutes. Wash and blot dry. Examine under oil. Acid-fast organisms stain red, and the background and other organisms stain blue. Medical application. In the clinical laboratory, the acid-fast stain is important in identifying bacteria in the genus Mycobacterium. Specifically, M. lepori, which causes leprosy, and M. tuberculosis, which is a causative agent of tuberculosis. That's all guys. For more videos, subscribe.